So we're going to look at some crosses done by Gregor Mendel. And he was a monk and he did work on pea plants and he looked at lots of different traits. So sometimes he looked at pea colour, maybe pea shape, also the height of plants, and things like where the flowers came from and what the flower colours were. But we'll have a little look at a cross he did to do with height of plants. So he took tall plants, bred them with short plants, and what he found was all the offspring were tall, 100% tall. So he took these plants and he bred them together. He said, I'll take the tall ones from this second generation and have a look at what we get. And what we get is some tall plants and some small plants, the short plants, in a ratio of 3:1. So 75% of the offspring were tall and 25% were short. Now let's have a look at what was going on inside the genotypes. So the parents' phenotype is tall, crossed with short. And these tall plants are big T, big T. So they are homozygous dominant, and the little plants are little t, little t. So we would call that homozygous dominant. That means that both of the alleles are the same. That's what homozygous means. And they are both the big T allele. They're the dominant allele. Over here, this parent is homozygous because the alleles are the same, but it's homozygous recessive because both of the alleles are little letters. They are recessive. And we know the recessive, they're about to disappear when we do a cross. So in our cross, what we use is a Punnett square. We have a square where we lay out the genotypes of the parents on each of the sides of the square, showing what the gametes are. Now, for this plant, the gametes are going to all contain a little t. So we put the allele that's present in the gametes inside a circle to show that it's in a gamete. And over here on this side, we put the gametes from this parent, and they're both big T. So all of the offspring in this first generation will have, when we recombine these gametes, when they fertilize each other, we will end up with an individual that has a big T and a little t. And that's actually going to be the same for all of these offspring. So each of these squares represents an offspring formed by the fusion of this gamete and this gamete for this square, this gamete and this gamete for this square, and you work it out for each of the squares. So in the F1 generation, all of the offspring are big T, little t, which is tall. Now we're going to take two of these individuals and breed them together. So we have a tall crossed with a tall, but this time their genotype is heterozygous. That means one big letter, one little letter. And when we draw out our Punnett square and show the gametes from this parent up here and from this parent on this side, the gametes are not the same as each other anymore because half of the gametes from this individual are going to be a big T and half are going to be a little t. Remember, in meiosis, we halve the chromosomes and we put half of the DNA into one lot of gametes and half into another. So we're going to separate the big T and the little t. The gametes from this parent are going to look like the gametes from these because they've got the same genotype. So Some of the gametes will have big T and the other half will have little t. Now, making our offspring is a little bit different. So in this square, we're going to fuse the big T and the big T. So our offspring here are going to be tall. And they're going to be tall, homozygous dominant. In this square, we have a little t and a big T. Always put the capital letter first. That's the convention. So it's going to look tall, but it's actually a heterozygous genotype, but the big T is dominant and the big T is tall. In this square, we've got a big T and a little t. Remember to put the capital letter first. So again, it's going to look tall. And the fourth option for offspring is a little t from this parent and a little t from that parent. So this time, our phenotype will be short because it's a homozygous recessive. And you can see how we got the ratio of 3 tall to 1 short in our F2 generation. So that's the grandchildren of the original cross that Mendel started with.